afternoon. Good afternoon, Madrid. I guess, well, I'm sure you are listening and you are viewing us, watching us. It's for me a pleasure to welcome you to the third edition of the Liver Seminars program, which was a program that Rafa Bañares and myself, we thought about it 4 years ago. And until now, we have been welcoming different speakers from around the globe to give talks about liver disease from a translational point of view. And today we start the third edition, and we are glad on that. Thank you all for being here. Just two things. We have a very complete program for this third edition. Today we start with Tom, but we have 8 more speakers coming from now until June. So you can check on our website all the speakers that will come to either to Barcelona or to Madrid. And of course you can also book a private meeting with the speakers to discuss science and new projects, etc. And finally, I would like to thank our sponsors, which are Gilead Sciences, the CIBER-ED, our CIBER, and the Spanish Association for the Study of the Liver for their great support. And that's it. Thank you, and Maria, I give you the word.
Good afternoon to everybody. Thank you, Jordi, for giving me this opportunity. It's an honor for me. It's a pleasure to introduce Professor Carlsen. You probably all know him very well. He was the previous secretary of ISEL, but also he's the research, as is written there, is the research head of the Division of Surgery and Inflammation and Transplant in Oslo University Hospital. He's also a full professor in internal medicine there, and you probably also know that his research is based on primary sclerosing cholangitis and mainly in genetics of the disease and microbiome. And we are very happy to have you here today. He is going to talk about the role of microbiome in all liver diseases. We asked him to talk about this in autoimmune liver diseases, but he preferred to give a more broad approach of the microbiome. So I hope you will enjoy the talk. Thank you very much for being here.
Thank you very much, Maria and Jordi, and dear audience. It's a great pleasure and honestly also a great honor to be here in Barcelona. You have a fantastic liver unit, worldwide known, and it's actually the first time I had the chance to visit this famous liver unit, and I must say that I'm not disappointed. So from my perspective today, I will talk a little bit around the gut microbiota, as Maria has alluded to. I know that there's a mixed audience here and also in Madrid, clinicians, basic scientists, translational researchers. So I will try to keep it at like a medium-high level, rather focusing on my reflections and my thinking in several of these areas than trying to plow through all the masses of data that we currently have in this aspect, both in relation to liver disease, but also other areas. In many ways, I mean, to put like a mark in the history when this all kind of took off and started booming, it's pretty hard. I mean, there's been a long-standing interest in the role of the gut microbiota in various disease states and human physiology. It goes back to the 60s and the 70s and even before of the last century. But in many ways, I think this paper that I'm showing you here kind of served as a landmark paper where you basically figured that by transferring the stool of this fat guy here versus transferring the stool of this lean guy here into germ-free mice, you had a totally different weight development and body fat development. And this was in 2006. I think that's the point that I want to make. This is a pretty recent history. And following this paper and in the following years, you had this blooming. This is in 2010. The history goes on for a little bit. You even start talking about the other genome and this physiological companion of us, and all really relating to this shift from the culture-based to genomics-based characterization of the gut microbiome. Very much is much of the developments in this field and the booming and this kind of shift that occurs in 2005, 2010, and the years to follow is related to the blooming and the development of genomic technologies, high throughput technologies. And this allows us in the lab, in the patients, in the stool, to characterize not only single bacteria, but the entire bacterial community of the bowel, not only of the bowel, but of the skin and all other human compartments, using these genetic technologies. 16S, you typically get down to family genus level. With a full metagenome sequencing, you get down to the species level, and then blooming in the kind of aftermath of all these developments, you have parallel disciplines, metatranscriptomics, metaproteomics, metabolomics, alluding to the fact that very much this is a big data discipline. And for us and the Oslo group, this came at the time when we had been doing big data for a long time. We did GWAS, we worked on human genetics. So for us, it was a logical shift to move all our technological skills and our big data handling capacities into this new domain and this new genome, the metagenome. So that was it for the history. And I think this is in many ways the key figure of my presentation, and I always like to start out by showing this for the mere fact that it's alluding to the point that when you're talking about the gut-liver axis, you get this illusion that it's something related to the gut versus the liver as a unilateral thing. And of course, there are many things going from the gut to the liver. We have all the nutrients, we have the microbial components, everything that we're exposed to through the oral exposures, drugs. You have bile acid reabsorption, you have immune cell trafficking, signaling molecules, etc. However, and I think that's the key point of this figure, there is also a reverse loop. This is a bilateral loop. There is also a liver-gut axis. Of course, this is where we are having our disease interests in the biliary tract. So possibly we have a particular interest in this, but there are the metabolites of a variety of endogenous and xenobiotic compounds. Of course, bile acid, phospholipids, cholesterol, immunoglobulins, and also signal molecules going the other way around. And then I think that's also part of the enthusiasm and the opportunities that open up here. This bilateral loop links then with the gut microbiome, with the bacteria. This was the old number. We have 10 times as many bacteria in the gut as we have human cells. This is the more recent estimate that we're probably quite equal. Number doesn't matter. It's a big number of cells within the gut, and all this is linked to the exterior, to the oral exposures, diets, antibiotics, drugs, even also other microorganisms, and of course including the exposures that we relate to every day in the clinic as hepatologists. In disease states, the microbiome is shifted, it's changed. And we can show this in a variety of ways, and it's shown throughout, I would say, the entirety of the human disease spectrum. These are just our own data. You can show that by a predictive capacity, where you see that distinguishing PSC from UC based on the microbiome is pretty easy. Here is early moderate NAFLD versus more advanced fibrosis, also pretty good utility. And then comes the question, and I think that's a fundamental problem that we're still struggling with, and which I will come back to throughout the presentation. How do we then know that these differences are not a result of a sick liver? How do we know that the disease processes per se do not alter the living conditions, the environment of these microorganisms in a way that they actually change, or these actually serve some potential causative pathogenic roles in disease development? And this chicken and the egg problem is inherent to all the findings that we deal with throughout this field. So it's shifted, so it's changed. And one very characteristic finding, looking at it at a very global level, all the microbes in total, is this reduced diversity. That means basically that you have less types of bacteria in your gut than you do in the normal state. This you see in NAFLD. These are pediatric patients. This you see in NAFLD-related phenotypes here, obesity, type 2 diabetes. This we see in PSC and in IBD. You can see that there is a gradient here from the healthy controls over to the patient sides in terms of diversity, and you also see it for fungi. This is alcoholic hepatitis. You see less types of fungi, less diversity in the diseased state. And unfortunately, somehow this is also happening to our planet. I don't know exactly whether, to what extent we should relate to that in our talk, but I think it goes back to the reflection on the chicken and the egg problem also, because of course, in a diseased organism, you lose out on expert, you lose out on those very specialized bacteria. They don't thrive in a diseased environment. On the other hand, you may say that, okay, so this reduced diversity per se holds pathogenic potential. And both may be true, but certainly if you compare this normal gut ecology to this disease state gut ecology, it's a totally different physiological machine. There are many things you can do here which you cannot really do anymore. And this has an impact on human physiology, which we will revert to. That was at a global level. Then if you look at the single bug level, you do your metagenome, your microbiota or metagenome studies, and suddenly then there pop up these single bug associations. This is just an example from our own first paper in PSC some years ago, where we had this finding of this Veillonella bacterium, which shows an increased frequency in PSC, and we became very enthusiastic. Is this the PSC bug, and what could this mean? There's this term, this pathobiont. We will talk about a few other pathobionts, which are basically like this guy down here. They are normally symbionts living in harmony with the organism, but could cause disease problems. However, this Veillonella that made us so enthusiastic, then came the papers in PBC, in autoimmune hepatitis, in HBV cirrhosis, in cystic fibrosis, in a variety of fibrotic states. They also saw this Veillonella, so obviously it cannot be the PSC bug. So what did it mean? Does it mean that this drug is then involved somehow in the fibrotic states of all these disease conditions? Or does it mean that again, the chicken and the egg problem, does it mean that the microenvironment of the bowel of these various disease states actually provides a niche where this guy really likes to be, so he thrives, he grows, and there becomes more of them? We don't know. We don't know yet. This is something we have to figure out. The biology of the Veillonella finding is not yet clear. It's certainly there in a variety of conditions, but it's definitely not the PSC bug. I think this summary slide from a review article in Gastroenterology now, online in November issue now, is quite illustrative. These are 48, they looked into 48 studies on the microbiome in IBD. So it's not liver disease, I apologize for that. The point is quite clear here. Even in that massive pool of patients, they struggled to find findings on the single bug level that actually exceeded a replication rate beyond 3 studies, which is quite amazing. So for the majority of the findings, there was basically no consistency at all across all these studies. For some of them, you were actually able to find 2. And again, you can actually recognize here Veillonella in the Crohn's pool. And for only this less than a handful of findings, there was more than 3 papers reporting on the same finding. And I think it tells you something about the insufficiencies of the 60ness and the species level characterization of the gut microbiome. There are technological biases to that, and certainly you're missing out on some key features of what's going on. And we will revert to that. How do we deal with that? This is another pathobiont proposal in PSC, Klebsiella pneumonia, that came out last year in Nature Microbiology. They found a lot of this Klebsiella in PSC patients, also some autoimmune cases and PBC cases, and also it has been reported in cirrhosis and some other conditions as well. We don't really see that in our population, and many of the other studies haven't really reported on this finding. So what's really the genuine role in terms, or the disease-specific role in terms of PSC, we don't know. What they do in this study, however, and I think that's the nice thing about this whole history, they go a bit further. They try to look into the biology of this Klebsiella finding. They look at its involvement, particularly in collaboration with two other microbes in Th17 activation. And also, and this was presented at AASLD this year, they use a bacteriophage, a virus that kills the bacterium very specifically, and see a murine signal in terms of disease amelioration. But again, the question is still there. We look specifically for Klebsiella in our patients, we see it mostly in the advanced cases, mostly associated with aggressive disease. So whether it's again a disease state phenomenon driven by tropism, basically that these bugs really like the diseased organism, or it's actually having a pathogenic role, I think the question is still out there. This is a fascinating paper which occurred just before Christmas. Many of you will have seen it. It's an alcoholic liver disease. In the background of all this lurks this other famous journal from a famous European liver society on Candida albicans. I will not talk about that, but obviously this is quite clear if you read this paper very carefully. It's a bit unclear where he got this inspiration for this cytolysine, and I think it came from the Candida story. But that doesn't matter. I think the key point, it's a beautiful story, it's a beautiful paper. They find that in the alcoholic hepatitis cohort that they investigate, they have about 80% of the Enterococcus faecalis. And what they also find is that 30% of these patients are positive for this cytolysine toxin. And furthermore, they see this clear dichotomy in outcomes between the cytolysine positive and the cytolysine negative patients with alcoholic hepatitis. As you can see here, if you have the cytolysine positivity, you do bad. Those are the alcoholic hepatitis patients that we all see, that we all fear, that do bad. And you have those who do better. And they also take this one step further. They use stool samples from the cytolysine positive and the cytolysine negative patients, put them into mice, and they see that the cytolysine positive, they have the hepatic features of the alcoholic hepatitis, whereas the cytolysine negative ones don't. And they use the same approach as has now been done for the PSC Klebsiella finding. They use specific phages to target the cytolysine positive samples before they put them into the mice, and they actually have then an amelioration of the phenotype. So I think this is so much for the pathobiome story. So you have these global shifts. You have those single bug findings. Some of them are interesting. We don't know all about the biology of these findings yet. So what else is there? How sure are we that this stool and the gut microbiome is important? I will mention fecal transplantation here. I will not talk too much about therapy because much of it is still in its infancy. Fecal transplantation is one of three very radical approaches that you can use to manipulate the gut microbiome. I already told you about this fat guy who drove the fat phenotype in the 2006 paper. I will talk about antibiotics, and I will talk about the super radical approach of colectomy, which sounds crazy, but it's relevant to our PSC patients. And I will try to convince you that as much as these are therapeutic options, they're actually putting an extra weight to the scale, telling us that the gut microbiota is important. It's more a proof of concept that the gut microbiome involves in disease than the future therapy. The first trials came with the metabolic traits. This is a Dutch study where you can see that the peripheral insulin sensitivity improved after fecal transplantation from lean donors, but not after autologous gut microbiota infusions. You have had several examples from hepatology as well. Hepatic encephalopathy, this is the long-term follow-up from the Bakay study, where you can see that the hepatic encephalopathy episodes go down in the fecal microbiota transplant cohort. They have less hospitalizations. So clearly for this metabolic phenotype, for hepatic encephalopathy, and for other phenotypes where this has been investigated, shifting out the stool for a short period of time. This is not a permanent solution. The drift back to your normal stool in a few weeks. There is a utility. However, there are not a 100% response. There are some responders and some non-responders. This is maybe the slide doesn't show perfectly here. But you can see that there is this responder group and there is this non-responder group. I think interesting, if you characterize the non-responders versus the responder group, you can see that probably within this responder group, there is more of this diversity problem that you saw in the outset. They have a sicker microbiota than the non-responders. And then, of course, it's logical that the microbiota transfer actually works. And also there is this complexity to the whole situation. Okay, so there is the recipient point, and then there is the donor point. I mean, we all work with liver transplant patients, so we know that there is something about the combination of the two. And also for the donor, you have these super donors that probably have some universal utility and some specific utility to the individual host. So there is a lot going on here. And then the question is, do you then use this? Is this the future of medicine? Stool transfer? I don't think so. I think this is at best an intermediate stage, probably more, as I told you, a proof of concept, something is going on there. And then we have to understand what is the biology of this? Why is this happening? And then I think in the next stage, we would have new therapeutics and new therapeutic opportunities. So what about antibiotics? Equally radical. You kill a lot of bacteria, just like with a cannon. And in PSC, this improves alkaline phosphatase. This is pretty clear across numerous antibiotics. Vancomycin improves alkaline phosphatase. Metronidazole improves alkaline phosphatase. Other antibiotics, rifaximin, etc. So then the question is, do you then provide your patients with the rest of their life chronic disease antibiotic prescriptions? I don't think so. As little as I think that the fecal transplantation is the ultimate solution here, this is not the ultimate solution. It tells you that the gut is important. Maybe we should probably have already known in PSC. But the key point is that we have to understand how and why do we observe these effects. And only by understanding this, we possibly would get to the biology that would allow us the new therapeutics. Even more radical. And then in PSC, you have this point that of recurrence, that after liver transplant, about 30, 40, maybe even 50% of the patients, they get the disease back. And the British group showed quite, I would say, dramatically that an intact colon, an intact bowel, increased the susceptibility of disease recurrence. When they expanded their finding in the entire UK cohort of their analysis, it wasn't that clear. However, if we look into our Scandinavian patients, we see exactly the same, and even with an increased inflammatory activity in the bowel, you see an increased recurrence rate. So then the question, as with fecal transplantation and antibiotics, is this the future of medicine of PSC? You collectomize all your patients. And maybe I'm making this point a bit dramatic now, but I think the key point again, no, you should not do that. What this tells you is that the gut is important. And then you have to understand why and how this is all happening. And this brings us to the kind of next stage of this presentation. Because I think what we have agreed upon so far is that in the diseased state, the microbiome is shifted. You can see that it's different, it's moved. The yellow is in a different place than the green part here. And it's smaller, so there's less diversity there. And those shifts, including the single bacterium pathobionts associations that I showed you, have quite profound impact on human physiology, human or the capacity of the microbiome in processing external compounds, and also human immunology. And what we're doing at the moment and what we're seeing at the moment is that this lack of utility of the species, maybe even genus level, bug characterization 16S methods in explaining to us what's happening here. Much is shifting at the moment towards metagenomes. This is really where we are at the moment. We do most of our stool characterization using metagenome sequencing, basically meaning that you sequence all the DNA that's in your stool, and then you have to puzzle it back together, which is a huge bioinformatic exercise, but perfectly feasible. And then you get out a different type of readout. You have gene abundance. So you have functional readout, and you can even compare this functional readout at the gene level with measurements, for instance, metabolomics, and you can check for consistency. Is this like, just a DNA analysis problem, or is it actually a true functional representation? The problem again, or the problem then that you run into with these types of assessments is that you have a huge number of findings. When you do these assessments, you have hundreds of biological pathways popping up, and then you have a problem in distinguishing where are the most important findings, what's really key to this disease, and again, this chicken and the egg problem. Is this just a phenomenon occurring in the context of disease, or is it part of the pathophysiological cause in this setting? What I try to do then, since you have these hundreds and even thousands of findings that you need to sort, I try to kind of categorize them, and we're struggling still with how to do this. This is just a very simple way of classifying, and these are just my inventions, so don't take them for anything, but a way of reflecting upon this. Let's start with the human physiology. That bacteria involved in human physiology has been long known. I mean, we know the K-vitamin metabolism involvement, and certainly we know the bile acid co-metabolism. Which is actually quite extensive, and it's expanding as we're now getting insight from the metagenomes into the functional capacity of the microbiome in bile acid metabolism. We're even discovering new avenues of potential pathophysiology. I will not go into all the bacterial metabolic pathways, but just allude to also that there are you get eye openers within this space. For instance, these oxo bile acids. There was this paper occurring now just before Christmas where they look into the role of these oxo bile acids, and this is actually quite a big class of bile acids. These are 21 molecules that we really don't know much about in human physiology. And they show in this Nature paper how these cells actually involve in the regulation of Th17 cells and Treg cells. And of course, for us working in a biliary disease, this was super interesting. Okay, so what's going on here with this microbiome shifts? You have the changes to the bile acid metabolism, and how does this all impact on immunology in the relevant compartments? So these are just examples. Another prototype example that you need to mention when you're talking about the gut microbiota in terms of bile acids is the signaling, where you have the FXR, FGF19 signaling pathway occurring both at the hepatocyte and the ileocyte level, FGF19 signaling through the FGF receptor 4, and where you have this clear regulatory impact of the gut microbiome through this secondary bile acids onto this the activity of this signaling pathway. This is very radically shown here with the germ-free mice, where you have basically very little signaling. FGF, for the clinicians, FGF15 will be the same as FGF19 in humans, where you have this negative feedback from the taurobeto muricolic acid onto the signaling pathway, which disappears once you introduce bacteria, because then this bile acid is metabolized and it no longer can exert its inhibitory functions. There's a lot to elaborate here in terms of physiology still. Clearly, this is also interesting from a therapeutic perspective. If you look at NAFLD and the immunometabolic regulation here, you have the phase 2 and the phase 3 data on the obeticholic acid, which is an FXR agonist, and you have also signals from the FGF19 analog NGM282. Similar signals you have in PBC and PSC, both at the FXR level with PBC and PSC, with OCA, the PSC paper never really published, and for the FGF19 level with NGM282, again, alk-phos signal in PBC. Where this will take us in these diseases in terms of true clinical efficacy remains to be seen, but at least it shows you that this compartment that you're dealing with here, which is interwoven with the bacterial co-metabolism, is likely to involve also in the clinical domain. So much for the endobiome. Then what about immunology? Also, of course, the gut microbiome interacts with the human immune system, both through specific peptide lipid antigens, through cell wall products and the innate immune systems, bacterial metabolites. And if you look at one of the key dogmas, I would say, in relation to liver disease, it's related to this gut leakage phenomenon, which occurs at increasing frequency throughout progressive liver disease, where you have increased systemic exposure of endotoxins and reduced microbial diversity, and again this influence on the physiological co-parameters here. Exactly what's happening here, I think that there is still a lot to be understood regarding gut leakage. I mean, is it just like a hole? I don't think so. I think these are quite complex and intricate mechanisms of molecular transport, molecular exposures. And I think we will have a kind of a bit more sophisticated way to describe this whole leakage phenomenon as this point evolves. Other interactions, these are mate cells reacting to bacterial metabolites, specialized T cells with conserved T cell receptors. So that also involves here, and also the adaptive immune system involves here, where you have here just an example showing the relationship between the dietary exposures, even very simple dietary exposures like glucose, onto the peptide antigenicity of a spectrum. This is a Science Immunology paper. I think the key point is just to show you that the immune microbiome interactions, they span all the spectrum from the basic innate mechanisms up to adaptive immunity. This is an interesting paper that came out just over Christmas. It's not liver disease, it's celiac disease, where they have this very interesting finding on this Pseudomonas subspecies as a possible candidate for why you get this strong HLA-related immunoreactivity against gluten, and that possibly as a processor of some of the adaptive immune responses that you see. I think that there is still a lot of debate around that topic of molecular mimicry. I'm not a particular fan of that concept, but there is something to this. And the key point again is that we have a huge reservoir of antigens potentially interacting, potentially cross-reacting with endogenous, with exogenous exposures. And finally, I had to show these papers on aspects related to immunotherapy, where you have checkpoint inhibition. These are 4 Science papers, quite convincing storyline on the impact of the microbiome on the efficacy of checkpoint inhibition. Pretty simple storyline can be essentialized from all these data, where you have the non-responders and the non-responders, and you're able to distinguish between them. You show some species level, also metagenome differences there. And when you transfer, and this is also a transferable phenotype, when you transfer these stool samples, which is quite popular exercise in these fields, you actually see that the mice behave in the same way as the host. For hepatology, this has become relevant over the last couple of years with the entry of checkpoint inhibition in our fields. The exact mechanisms here, the exact cellular mechanisms even, are still to be elaborated. And I think whilst we might, and we discussed that earlier in the meeting today, whilst we might contemplate the utility of fecal microbiota transplantation, I think that, again, you're back to this point about understanding why this happens, how does this happen, and then I think we would potentially be we would be put up with new tools for changing non-responders into responders in this very important territory of liver oncology. Final point regarding the immune system interactions. This is, again, not liver field because we don't have any good data there. This is a Cell paper which had a lot of attention last year on pancreatic carcinoma, where they look at the tissue microbiome, because obviously you have the gut microbiome, you have the bugs in the lumen, then you have the mucosa, where you have mucus and a lot of tight interactions. But then there are also the sort of supporters. There has always been this group of people within this field that believe in the tissue microbiome. I'm a bit skeptic to this whole story, I have to admit that. But these data are quite convincing, particularly in this neoplastic setting, where they show that, again, linking predominantly with diversity, but also for single bug associations, with the outcome of the patients, again, relating to the level of immune activation in these patients. So that was some surveys through the immunology. And then what about the exposures? Everything we put into our mouth, as we showed in one of the opening slides, at some point get exposed to these billions of bacteria and their enzymatic machineries, their physiology. And there are numerous examples also from existing medicine where this is important. Everyday gastroenterological practice, and we will revert in a minute to this TMA example. This is fatty liver disease. Again, one of those characteristic papers where you see a lot of bugs going up and down, and then, as I told you, for IBD and all these other stories, most of them will never replicate in any other paper. The point being still that if you look into the biological outcome here, the functional potential of all this, it can be categorized in some key avenues. We already talked about the bile acids a little bit. I will mention quickly here TMA and the short chain fatty acid aspects, because I think that they illustrate some general principles of working in this territory. The TMAO story arrived in 2011. There were a Nature paper, a Nature Medicine paper, and a New England Journal of Medicine paper from Stan Hazen's group, where he showed that carnitine and choline from meat, from cheese, fish, etc., could be processed into this atherogenic compound TMAO. However, only in the presence of the gut microbiota. Interestingly, and this was 2011, as I said, this is 2013, the TMAO story was alluded to even before, back in 2006, looking into fatty liver disease. And you can see that you have already at that stage TMAO up as a pathophysiological factor in fatty liver disease in this PNAS paper back then. And probably, I mean, you never know. I didn't talk to him about it, but probably Stan Hazen knew about these findings when he was dissecting his screening, because he was working from an omics perspective. He did metabolomics. He found a lot of compounds that potentially associated with atherosclerosis in his cohorts. And then he selected somehow these TMAO related compounds. I think the key point is what he did next, Because what he did next, he did a lot of experiments, quite simple experiments, but still interesting. He saw that only upon oral exposure you would have this generation of the TMA, TMAO. If you gave the choline intraperitoneously, you wouldn't really see this this TMAO generation. You can see TMAO is flat here. And then when he gave it through the oral route, he had the TMAO generation. Then he gave the animals antibiotics. The TMAO production disappeared. And then they were conventionalized. They basically had their bugs back. And the TMAO production came back as well. And I think this tells us a little bit about how we're now drilling into these phenomena and understanding not only the bugs, the name of the bugs, the genes, etc., but also how this physiology works. It's basically a physiological expansion here, and I think what we realize is that biochemists, chemists are becoming very useful partners for us in these experiments. The other story is also clearly alluding to the involvement of biochemistry. I mean, you need people to work on this. This is the generation of butyrate. This is the generation of propionate, short-chain fatty acid from fibers. Fibers being a key nutritional factor under a lot of scrutiny over many, many years. It has been known for some time that there are some bacteria that do produce these short-chain fatty acids and others don't. This is known from the culture periods. You don't need metagenomes and blah, blah to figure that out. And you can see a lot of old literature cataloging bacteria on the basis of their capacity to produce butyrate and propionate. What has this got to do with disease? This is just one example. There would be numerous examples. I find this interesting. This is a cohort of best diabetic care, so to say, the red one here, and best diabetic care plus fibers. And you can see that the impact here on fibers is quite profound on HbA1c. And what they are doing in this paper, and I cannot show you all the data, they're able to link this to butyric acid production. And they're even going further from that into candidate high butyrate producer bacteria, which they're actually able to show ameliorates the metabolic consequences of high-fat diet. Here they take this one bacterium here, add it to the high-fat diet, and you can see how this high-fat diet exposure in these animals gets less of an impact, illustrating the possible functional capacities of butyrate in this system. And I could go on and on. I could take one compound, one dietary compound, one whatever drug after the other, and we would be late into the evening. It's already getting late. So I think that the key point with all these kind of exposures that we have, all the things that are coming from the gut, I think that the model that I want to illustrate and that I think we need to be working at the moment is to consider this big physiological machine here in the bowel as a companion, as a physiological companion to the liver and to the host, and only by understanding the details of this physiological machine, we will be able to understand how diseases which are closely linked with this physiological machine, for instance liver diseases, actually develop. And this also, of course, include drugs. This is an interesting example. I keep loving this paper in C. elegans. Why that? Because these are very basic scientists, and they write this very cute thing in the introduction of the paper. When you read the paper, they tell us that they found this drug very interesting because it gave bowel disturbances. And as doctors and gastroenterologists, which drug does not give bowel disturbances? So, but anyway, that's what they propose, and they did a lot of working on metformin, basically showing how this involves in the mechanism of how the gut microbiota involves in the in the metformin pathway, and this led to a lot of nice stories alluding to this partnership of the gut microbiome into drug function. The key point here being that when we're talking about drugs at the moment, we're talking about drugs that are going and working against the host-targeted drugs, and we're talking about drugs that are targeted against the bacteria, mostly at the moment still aiming at killing them. That's what we're interested in. We have antibiotics, and then we have the host-targeted drugs. And I think what I'm trying to allude to here is through this deep dissection that is currently ongoing of this physiological companion, I think we will get a little bit away from this dichotomy of either you kill the bugs or you help the host, and into a paradigm whereby you would specifically target these biochemical pathways that are emerging in front of us, and that this, in the end, possibly could lead to new therapeutic principles and therapeutic efficacy in patients with liver disease broadly. So that's all I was planning to say today. This is in Oslo. This is the group in Oslo. You cannot really see their faces very well because we have so nice weather in Oslo. And this is my son, and you probably all recognize where he is. So I don't have to say that. And I will just leave my take-home messages up there. I saw some of you took pictures. You can have the slides. I mean, I never keep my slides to myself, so you can just email Jordi or Maria, and you will have the slides, and you can use them as you like. So thank you very much for the attention, and I think that there is also time for some discussion now. Yeah. If I were to put my money onto any kind of single hypothesis at the moment, I certainly think that there is something going around in the enterohepatic circulation of bile acids that drives the disease progression. If you look at the disease distribution, you have the biliary inflammation, you have the right-sided predominance of the inflammation, even some ileal inflammation. So it's all occurring throughout this enterohepatic circulation, whether that is a peptide, whether it's a small molecule, whether it's a bacterial metabolite de novo, whether it's xenobiotically transformed compound, we don't know. And I think screening for all this, I mean, the metagenomes are quite easy. Those are easy analysis. And endogenous metabolomics is also quite, I would say, advanced. When you're starting to look into, xenobiotics and exogenous, the metabolomics of the exterior, you end up with virtually, I would say, limitless number of potential candidates, and how to use the knowledge that we're currently gathering from the microbiome into narrowing down the number of candidates, narrowing down the number of potential primary causative events. We're still learning, but I definitely think that there is some causal factor relating to the gut microbiome. Yeah. The point is that between the PSC patients, either they have IBD or not, there is a difference with ulcerative colitis. And I think from a general perspective, we don't really see the IBD of PSC as neither ulcerative colitis or Crohn's disease. I typically often talk about that. I didn't show any genetic data. We see it also from the genetic studies. So I think it's just a totally different disease than ulcerative colitis. And there are many commonalities, even if the patients have already inflammation. Of course, the inflammation, as it gets more severe, introduces a bias to the microbiome. I would hope PSC, but I mean, I'm I wouldn't say that. Honestly, I think, I mean, we are already doing it. I think that's the point that I alluded to already with the FXR, FGF19 analogs, etc. You're somehow already into this gut-liver axis. Whether you're then talking about microbiome-related therapies as such, I think the volume of research and the pressure is much higher in the metabolic territory, where it goes far beyond NAFLD. I mean, all the research happening in type 2 diabetes in cardiology. So probably they will win the race, and that's where we will have the first applications. But these are mere speculations. I think it's still in its infancy exactly what these treatment modalities would look like. Would you talk about, as I mentioned towards the end, drugging drug the bugs in terms of providing an interference with their physiology? Would you talk about using the bugs, modified bugs, microbial components, genetically modified bugs as drugs at the other end? Whether that will really be the utility that we would be seeing, I'm not sure, and I think this all has to be understood before we can talk about any disease, because as I said, I think that these crude, gross approaches of antibiotics and fecal transplantation, they're in the intermediate stage that we should use as part of elaborating the mechanism. They're not the penultimate. It's not the future of medicine. So, but if you ask me and push me to answer that question, I think possibly we should say it's in the metabolic space, meaning NAFLD. Yeah. No, I think it's really a major confounder in all these studies, and I think that if you look at it in that way, so let's stay with the luminal microbiome, not talk about the mucosal microbiome, which is a story on its own, not talk about these speculations about the tissue microbiome. If you talk about luminal microbiome, certainly transit time and many things that would influence the final status of what you collect in your studies. Diet, profound impact. Particularly outlier diets, vegans, etc., would totally, mess up your data. And also the tools that we have for collecting food data, I would say, are as complex. I mean, we have some tools. Food frequency questionnaire is often used, and there are even more elaborate tools, but they are all kind of, and this changes throughout the day. And I think what the bottom line here is, yes, there is a lot of bias to the sampling collection. And I think what we experience hands-on with that is that what we thought was a well-powered study with a few tens of individuals, maybe even 100 or 200, it's not really enough. There is so much noise in these systems to have really the clear findings standing out. You're probably talking hundreds, possibly thousands, probably thousands of samples. And only at that level you will start kind of looking above the intra-individual and the inter-individual noise. So I think that much of the data that has been coming up, it's a bit reminiscent of the early days of genetics, where all these candidate genes came out every week and they were all false, we know, in retrospect. And I think that's the case for much of the findings that are published here as well. They're just chance findings, and most of them will not remain. No, most of these studies are cross-sectional studies, and as I already mentioned in the comment to Jordi, most of them probably reporting mostly spurious associations that will not remain. I think what we try to do, and what we have transformed most of our studies, we're doing longitudinal samples, so you have, of course, more samples over time, so you try to get into the core of something constant. We also work with interventions, because I think that when you have specific interventions, drug interventions, proof of concept intervention trials, you would gain some insight, knowing the mechanism of your interventions. We did a study on statins, for instance, looked at the microbiome and trying to elaborate that. But it goes through the route of even then putting an analog with the genetic studies. We found all these genetics in the GWAS era, and then now we're still struggling to figuring out what they all mean. And I think that's the case here as well. You will have those associations. You have to do your basic lab work. You have to take these findings, those bugs, the metabolic pathways that they allude to, into your mouse, into your cell systems, and explore and elaborate. And it will not come easy. And I totally agree with you. We're at the very, very cross-sectional stage of this whole undertaking. And it has to be refined, and it has to be taken through this big functional exploration, which will take and will keep us busy for the next decades, I think, to figure out the whole of this physiology and how it relates to liver disease. Yeah. And I fully agree. And I mean, of course, it was a special case, this sepsis case, antibiotic resistance. I mean, in patients, they do get sepsis. And but I perfectly agree. I think that was my key point regarding fecal transplantation, antibiotics, and also colectomy for PSC patients. They prove that the gut is important, but that's not the way we're going to treat patients in the future. We have to figure out how, and it's this how stage which I think is coming up now. Now we know that there is a problem there and that there's a potential of a vast resource of knowledge, and now we have to go and get it. Yeah.
On time, we can finish here. Thank you very much for being here today. It's been a pleasure, and we enjoy a lot your talk. Thank you very much.
Thank you.